This video is the first lecture video among like 4 videos that I will post for the genetics module. So for the entire module, we will talk about Mendel's law of segregation, the law of independent assortment, and then pedigrees and inherited diseases. So this first set of objectives will actually extend into the second lecture video. But for this lecture video, we are going to go over the terms you see on number 1 here. And then we will move into law of segregation, Punnett squares, and then using Punnett squares to determine phenotypic and genotypic ratios. So we've talked a little bit about alleles when we were discussing mitosis and meiosis. So this will serve as a review. So the technical definition of an allele is the version of a gene. So I've been using eye color in a lot of my examples, so I will continue with that for now. So if we are talking about alleles, an example would be the blue eye allele and the brown eye allele, okay? So for many of our genes, we have 2 different alleles. Now I say many of our genes because there are some genes that have more than 2 alleles, like the gene that codes for blood type, for instance. Now, everybody or every organism has at least 2 alleles per gene. You inherit 1 allele from your mother, 1 from your father. So remember when we talked about eye color gene, I believe I used brown eyes for mom. So you would inherit the brown eye allele from mom, the blue eye allele from dad. I'm going to summarize that for you here. So I say every organism, it's actually like organisms that reproduce using sexual reproduction. So every organism has at least 2 alleles for every gene. 1 allele is inherited from mom and 1 allele from dad. Now, these alleles do not have to be different, but a lot of times they are. So let's look at the image here. Here we have 2 chromosomes, and these are homologous chromosomes. So they make up a pair, and they code for the same genes, but they can have different alleles. Remember, homologous chromosomes are not identical, but they code for the same genes or traits. So here we are looking at chromosomes in a plant. So one of the chromosomes has the allele for purple flowers, 1 has the allele for white flowers. The gene is flower color. So right here, it says locus for flower color gene. Locus means the location on the chromosome, right? This is set. Genes don't usually move around on the chromosomes. They are set in their location. So we call that the locus. So now we are going to talk about some of those terms that I gave you on the first screen. And I'm also going to explain dominant versus recessive alleles. So here we have flowers from pea plants. Now, most things that you read about genetics will start with this example because pea plants are what Mendel experimented on. Gregor Mendel is considered like the father of genetics. He was a monk in Austria, and he conducted many experiments to determine how traits are inherited. So there are purple flowers and white flowers in these pea plants. So we are looking at the flower color gene. Trouble moving that toolbar. There we go. Okay. So when you are looking at an individual organism, it will have at least 2 alleles per gene. Now, an individual that is homozygous for a gene means that that individual has 2 of the same alleles. So that individual would be big B, or little b for the flower color gene. So actually, let's take a step back here. So we use letters to symbolize an allele, okay? Now, when you are looking at a gene, you have to use the same letter. So in this example, the letter is B. So a letter is used to represent the alleles for a gene. So the same letter is used for the alleles. So in this example, the letter B, capital B, represents the purple allele. Lowercase b represents the white allele. Okay. So let's revisit this definition here for homozygous. So a homozygous individual has 2 of the same alleles. So homo means the same. So a homozygous individual, homozygous for that gene, will be either big B, or little b. Now, an individual that is heterozygous for a gene means that they have 2 different alleles. So that individual would have to be capital B, lowercase b. Okay. So homo means the same. So a homozygous individual for a specific gene is big B, or little b. An individual that is heterozygous for a gene will have a capital B, lowercase b, meaning they have an allele for purple and an allele for white. Big B means the individual has 2 alleles for purple. Little b means 2 alleles for white. So the question that I put on this slide asks you, when are recessive traits visible in the phenotype? So prior to answering this question, I'm going to talk about the difference between genotype and phenotype. So genotype. The genotype of an individual refers to the alleles that they possess for a gene. So you could think of it like the letters, okay? So an individual can be big B, big B, little b, or little b, little b. Those are 3 different genotypes. So for genotype, I'm going to write the alleles that an individual has for a specific gene. So examples of the genotype would be big B, big B, big B, little b, little b, little b. So when you are doing genetics problems with like Punnett squares, the letters are the genotype. And I will explain in just a minute why we have to differentiate between genotype and phenotype. So I always think of phenotype, physical appearance, right? They start with a PH. That's how I remember things, but whatever works for you. So phenotype is the physical appearance of the organism for the specific trait. Okay, so in the example on the right here, it would be purple or white. Okay, the flower is purple, it looks purple, or it looks white. Now, the genotype does not, or the phenotype, let me say, does not necessarily tell you what the genotype is of the individual. So if you look at the image here, I have 2 types of purple flowers, or purple flowers with 2 types of genotypes. So here we have big B, and that gives us purple. Big B, little b will give us purple. So if I'm just looking at a purple flower, I'm looking at the phenotype, I do not necessarily know the genotype. It could be homozygous dominant or heterozygous. Now, if I am looking at a white flower, since white is the recessive allele, then I do know the genotype. It has to be little b, little b. So let me elaborate on that here. I'm going to erase some of these text boxes that I put here, so it looks a little more organized. Okay, so I mentioned the terms dominant and recessive. Now these refer to the alleles. So if you have a purple allele and a white allele, one of those alleles is dominant, one is recessive. Now, this only applies to your straightforward Mendelian genetics. There are many exceptions to this, but we are not going to go into that. So we have a dominant allele. So what this means is if you have a heterozygote, then the appearance of that heterozygote tells you which allele is dominant. So if you look at my picture, big B, little b, it's a heterozygote, shows up purple. That tells me that the purple allele is dominant. So this is how I'm going to define it. The dominant allele masks or hides the physical, let's see, how do I want to say, masks or hides the physical trait of the recessive allele. So the example, in the heterozygote, big B, little b, the purple allele hides the expression of the white allele. Okay, so with big B, little b, the flower is purple. There is no white. The purple allele is dominant and it is hiding the expression of the white allele. So the white allele in this case is the recessive allele. And what that means is the physical appearance of the organ of the, we'll say, of the recessive allele is only present in an individual with two recessive alleles. See if I can explain that better with an example. So, white is the recessive allele in this case, right? So the flower will be white only in an individual with little b, right? Two white alleles. If there is a capital allele, well, one second here. So if there is a capital allele or the dominant allele present at all, the flower will be purple. So if you look at these individuals, if there is a capital B in the genotype, the individual plant has purple flowers. The only way it will have white flowers is if the genotype is little b, little b. So that is the difference between a dominant allele and a recessive allele. And the only way you can tell which is dominant, which is recessive, is to look at the appearance of the heterozygote. Okay. Okay. So I'm going to go back to these definitions that I asked you to know. And I am going to summarize them for you again, just to make sure that you understand them. So let me switch over to my questions. Okay, you should be able to see my PowerPoint with the questions. So these are the terms that I gave you on that original slide with your learning objectives. So let's go through these to make sure that you understand them. So the dominant allele, I'm going to define this a little bit different than I did just a few minutes ago. So the dominant allele determines the phenotype if it is present. So what that means is if, say, the plant has a big B in its genotype at all, the flower color is purple. So big B, or big B, little b. So big B, or big B, little b flowers are purple. Okay, there we go. So a recessive allele So what I'm going to put for the recessive allele, it is masked by the dominant allele. So the recessive allele, let's see, only determines the phenotype in a homozygous recessive individual. That would be little b, okay? So big B is homozygous dominant. Little b is homozygous recessive. Now, with heterozygous, you don't say dominant or recessive because you have a dominant allele and a recessive allele. So remember, heterozygous, hetero means different. So the individual has two different alleles for a gene. So big B, little b. Homozygous, the individual has the same allele for a gene. Now, it could be big B, or little b. So for homozygous, you can specify homozygous dominant, homozygous recessive. Now the genotype. The genotype indicates the alleles present for a gene. I'm going to write the alleles present in the individual for a specific gene. So these are the letters. So you would have big B, little b, little b. Those are all genotypes. The phenotype is the physical appearance of an individual. And I'm going to put for a specific trait. Trait, gene, I kind of use them interchangeably, but trait would be like, you know, eye color. And then there's the eye color gene, so they're kind of the same. So in this case, the trait is flower color. So the physical appearance can be purple or white. Now remember that for the dominant color, which is purple, you do not know if the individual is big B, or big B, little b. So this is a short video as an overview to genetics. In my next video, I will talk about the law of segregation and we will look at Punnett squares.